So you want to clone your dog, not adopt the same breed, not find a puppy with similar markings. You want a new animal created from living cells taken from the one you already loved. The public price in the United States is $50,000 for a dog and $50,000 for a cat, split into 2 equal installments. But the first payment can be much smaller. A company can charge $1,600 to grow and freeze a line of your pet's cells, then $150 each year to keep those cells stored. The expensive decision can wait 5 years, 10 years, or longer. The $50,000 clone gets the headline. The stored possibility gets most customers through the door. Under Viagen's current cell storage agreement, the initiation fee and annual storage fees paid for that cell line are applied toward the future cloning fee if the customer moves forward. So the commercial ladder is not years of fees plus another $50,000 on top. It is a staged path toward the $50,000 cloning service. The clone gets the headline. The stored possibility gets most customers through the door. And even when the science works exactly as promised, the company cannot return the years the animal already lived. It cannot transfer memories, training, household routines, or the personality that emerged from them. It can produce a younger genetic twin. It is extraordinary. It is not resurrection. One customer profiled by The New Yorker shows why someone still pays. Retired police officer John Mandola preserved cells from his dog Princess during cancer treatment. After she died, he waited months before ordering. He eventually received 2 puppies, Princess Ariel and Princess Jasmine. They strongly resembled Princess, but not perfectly. Their golden markings differed slightly, and each puppy had a misaligned eye on a different side. Mandola had not purchased a rewind button. He had purchased the closest biological continuation available and the chance to build a new relationship with it. By the end of this video, you will understand how pet cloning works, why the first sample can become urgent, how the company earns money before a clone is born, why early payments become future credits, why one delivered puppy hides many biological attempts, and how a $50,000 price can still produce a fragile margin. Because this business is not selling the same animal twice, it is selling genetic continuity, delayed commitment, and a second chance to meet a later-born twin. Before talking about the business, we need to define the product correctly. Pet cloning is not selective breeding. It is not gene editing, and it is not a process where a veterinarian takes DNA, prints an animal, and installs the original personality back inside it. The commercial method is called somatic cell nuclear transfer. The name sounds complicated. The basic idea is not. A laboratory takes a living body cell from the animal the customer wants to clone. That cell contains a nucleus carrying almost all of the animal's genetic instructions. The laboratory also needs an unfertilized egg from a donor animal. The original nucleus of that egg is removed. The nucleus from the customer's preserved cell is placed into the egg. The reconstructed egg is activated so that it begins developing like an embryo. If development continues, the embryo is transferred to a surrogate mother who carries a normal pregnancy and gives birth. The puppy or kitten receives the original pet's nuclear genome. It does not receive the original pet's brain. A clone can reproduce nuclear DNA. It cannot reproduce a life already lived. The egg still contains mitochondrial DNA from the egg donor, so the clone carries the original animal's nuclear genome rather than being identical in every biological molecule. It will be the same sex and can closely resemble the donor in body structure, coat, and inherited tendencies. But genes are instructions, not a recording. Identical twins can share almost the same nuclear DNA and still become different individuals. Here, the twins are also born years apart into different environments. The simplest, accurate description is this: a cloned pet is a later-born genetic twin. That definition matters economically because it sets the limit of the promise. A company can verify genetic relatedness with a laboratory report. It cannot certify that the new puppy will choose the same chair, fear the same vacuum cleaner, trust the same people immediately, or respond to the same nickname in the same way. The company has to explain that limit before taking the money. And before any of the science is possible, it needs something the customer may be losing quickly: living cells. If a pet is healthy and alive, genetic preservation can be planned. A veterinarian collects a small tissue sample. The sample goes to a laboratory. The lab attempts to grow living cells from it. If the culture succeeds, those cells can be frozen in liquid nitrogen and held for future use. If the pet dies unexpectedly, the situation changes. Viagen's current emergency protocol says that under optimal post-mortem conditions, with the tissue kept cool at approximately 4 degrees Celsius and not frozen, biopsy samples can be accepted for up to 5 days after death. It also warns that every passing day reduces the chance of successful preservation and that a sample from a living animal is preferable. That creates one of the most unusual sales environments in any consumer business. The buyer may be grieving. The product is unfamiliar. The clock is real. And the first decision can preserve an option without committing to the full $50,000 service. That is why the lower-priced branches matter. Express Tissue Banking, currently $500, stores tissue without first proving that a viable cell line can be grown from it. Full Genetic Preservation, currently $1,600, attempts to establish and freeze cultured cells. Viagen says the customer is generally notified of the result within 4 weeks. The first product is not a puppy. It is an option that biology may close forever. The commercial distinction is simple. Tissue banking stores a chance. Genetic preservation attempts to store a usable input. Cloning uses that input. The cloning guarantee begins only after a viable genetic sample exists. Viagen currently advertises a full money-back guarantee on the cloning service if it cannot deliver a healthy clone from a viable sample. The genetic preservation fee is separate, and the company's current storage agreement expressly disclaims any guarantee that stored cells will ultimately produce a clone. So the first transaction is not a miniature clone order. It is option creation. The customer is paying to prevent biology from closing a door before they know whether they ever want to walk through it. That makes veterinarians critical distribution partners. The company does not need a retail laboratory in every city. It needs local practices that can collect and move a sample quickly. Viagen says its veterinary network has more than 500 members. And once the cells are safely stored, the company can keep the customer relationship alive for years. Now the pricing structure becomes easier to see. Express Tissue Banking costs $500. It freezes the tissue itself without first confirming that a viable cell line can be cultured. Full Genetic Preservation costs $1,600. The biopsy kit is included when it is used and returned. Viagen retains $300 only when a kit is sent but not used. Annual storage is $150, beginning on the anniversary of the agreement. And cloning is priced at $50,000 for a dog or cat. State sales tax, outside veterinary work, and some shipping costs can add more, but the preservation and storage payments are not automatically added on top of $50,000 for a customer who later clones. Under Viagen's current agreement, those payments for the same cell line are applied toward the cloning fee. Take a customer who preserves cells and stores them for 10 years. The initiation fee is $1,600. 10 annual storage payments add $1,500. Total paid before cloning, $3,100. If that customer later initiates cloning, the simplified remaining balance is $46,900. The lifetime amount paid to Viagen across preservation, storage, and cloning remains approximately $50,000 before tax and outside costs. If the customer never clones, the $3,100 is the entire commercial relationship. For customers who clone, preservation payments are credits, not extra charges on top. For a non-converting customer, preservation and storage are the final products. For a converting customer, they are also credits against the high-ticket service. So the value of storage is not the ability to collect years of fees and then charge a second full cloning price. Its value is timing, retention, and option control. The company receives money before the hardest biological work begins. It keeps custody of an irreplaceable cell line, maintains the relationship, and builds a pool of potential future cloning customers. A 2025 American Animal Hospital Association article quoted a Viagen manager saying the vast majority of customers purchase genetic preservation rather than proceed directly to cloning. That detail changes the business. The broad offer is the option to decide later. The high-ticket offer is the attempt to exercise that option. A $50,000 decision can feel impossible during a crisis. A $1,600 decision can feel like buying time, especially when the payment is credited if the customer eventually converts. The customer is not saying yes to cloning. They are paying not to say no forever. But at the top of the ladder, the business stops looking like data storage and starts looking like a biological factory with an unpredictable production line. Imagine running a manufacturing company where the raw material is a preserved cell. The production equipment includes living donor animals and surrogates. The cycle takes months, and the final product cannot be remade by simply adding an overnight shift. That is the cloning operation. First, the stored cell line has to be viable. Then the laboratory needs suitable donor eggs. Each egg has to be prepared. A donor cell nucleus has to be transferred. The reconstructed egg has to begin dividing. The resulting embryo has to be viable enough for transfer. A surrogate has to become pregnant. The pregnancy has to continue to term. The newborn has to be healthy. Then the puppy or kitten goes through an ordinary nursing period before delivery to the customer. Viagen says gestation takes roughly 60 to 65 days and nursing another 56 days. Those biological clocks start after the customer has already waded through cell preparation, scheduling, and embryo work. In March 2025, the American Animal Hospital Association reported a 5 to 7 month wait list for the company's pet cloning service. That figure should not be treated as a current guaranteed queue. Viagen's present FAQ says only that timelines vary with demand and orders are handled in sequence. The important point is that capacity is not measured only in laboratory benches. It is measured in suitable eggs, synchronized reproductive cycles, veterinary staff, surrogate availability, animal care space, and time. The customer pays the cloning price in 2 equal installments, half to begin and the remaining half when a healthy clone is delivered. You can expand a laboratory. You cannot add a night shift to gestation. Viagen currently advertises a 100% money-back guarantee on the $50,000 cloning service if it cannot produce a healthy clone from a viable sample. The signed contract controls the final terms. But the public promise clearly places a major part of technical failure on the provider. The customer risks time and emotional disappointment. The company risks labor, animal care, laboratory materials, and capacity that may never produce completed cloning revenue. This is why the $50,000 price cannot be read as a simple markup on a puppy. The visible animal is the successful endpoint of a much larger sequence. And unlike an ordinary factory, the company cannot fully standardize the biology entering every step. Breeds and cell lines can behave differently. Donor cycles do not arrive on a spreadsheet, and pregnancies do not shorten because the quarterly target is behind. Records and protocols can scale. Gestation cannot. That makes the revenue look large and the throughput surprisingly constrained. From the outside, pet cloning looks like one luxury transaction, one customer, One laboratory, one $50,000 animal. Inside, the commercial stack is broader, but the layers have to be counted correctly. The first layer is genetic preservation, the entry product, the scientific prerequisite, and, reportedly, the service most customers purchase. The second is storage. The annual payment repeats while the client waits. For customers who never clone, preservation and storage are the final revenue. For customers who convert, those payments reduce the remaining cloning fee. So, storage is earlier revenue, retention and demand visibility, not free additional lifetime revenue. The third layer is cloning. It produces the headline number, but the incremental invoice can be lower than $50,000 after prior credits. It also consumes the most biological capacity and carries the public money-back promise if a viable sample does not produce a healthy clone. The fourth layer is the veterinary network. Local practices collect and move samples, giving the company national reach without building a consumer clinic in every market. The fifth is the technology platform around the pet service. Viagen has long worked in horses and other animals. In November 2025, Colossal Biosciences acquired the company, emphasizing assisted reproduction, cryopreservation, biobanking, and species preservation. The pet business continued, while the same capabilities became part of a larger conservation platform. The consumer clone may be the most visible use of the technology. The reusable asset is the process underneath it: cell culture, cryogenic storage, reproductive coordination, veterinary protocols, and quality systems. Storage creates retention and earlier cash. It does not create a second full cloning fee. The American Pet Products Association says 95 million U.S. households owned a pet in 2025, and industry spending reached $158 billion. But cloning serves the tiny intersection of owners who are emotionally interested, financially capable, able to preserve viable cells, and comfortable with the limits of the result. This business does not need millions of cloning customers. It needs a broad preservation funnel, a durable storage relationship, and a small number of people who eventually move to the top. The customer pays because the genetic outcome is emotionally scarce. Whether the provider earns a margin depends on how many invisible biological attempts that outcome consumes. And that brings us to the denominator nobody sees. A customer sees one puppy. The laboratory sees the denominator behind it. When the first clone dog, Snuppy, was created in 2005, researchers transferred more than 1,000 reconstructed embryos into more than 100 surrogate animals. Only a small number of pregnancies began, and one puppy survived to adulthood. That historical result is not a fair description of current commercial performance. The science has improved dramatically. A 2022 study analyzing 1,000 cloned dogs reported live offspring efficiency above 2% when measured against transferred reconstructed oocytes. That sounds low because it is low. But the denominator matters. It does not mean a paying customer has only a 2% chance of receiving a clone. A commercial program can use multiple reconstructed oocytes, transfers, and cycles for one order. The company-level success rate, the embryo-level success rate, and the customer order success rate are three different numbers. Marketing shows the customer outcome. The cost structure is built around the attempts. Unsuccessful eggs, transfers, pregnancies, and delays still consume labor, materials, animal care, and calendar time. Viagen's website also displays an 80% cloning efficiency claim without defining the denominator on the same page. It may refer to a valid internal project measure, but it cannot be compared directly with a scientific paper measuring live puppies per transferred reconstructed oocyte. In this business, the denominator is part of the economics. If the operator can improve embryo development, reduce repeated cycles, coordinate donors more efficiently, or predict which cell lines will perform well, it can complete more paid projects using the same physical infrastructure. A few percentage points of scientific improvement can be worth more than a large marketing campaign, because demand is not the only bottleneck. Biology is inventory, and the inventory cannot be ordered from a wholesaler when the shelf is empty. The same issue explains why a company can have a queue even when the number of final customers appears small. 20 or 30 active cloning projects can represent far more than 20 or 30 laboratory events. Each project sits inside a sequence of attempts, pregnancy windows, and animal care obligations. The customer buys one outcome. The company pays for the attempts required to reach it. And after all of those attempts, the company still has to deliver something the customer may misunderstand before it ever arrives. A new animal with old genes. This is the most important non-financial risk in the entire business. The customer may understand the word clone scientifically and still feel the word emotionally. Scientifically, the animal is a genetic twin. Emotionally, the purchase can feel like continuity with one irreplaceable companion. Those are not the same promise. Viagen's own FAQ says a cloned pet can show the same temperament, intelligence, and appearance, while also explaining that environment influences personality and behavior, and that markings may vary slightly. That is the honest tension. Genes influence body shape, coat, sex, inherited tendencies, and many parts of behavior. But a pet's personality is also built by prenatal conditions, early handling, socialization, training, household routines, other animals, illness, accidents, and thousands of ordinary interactions that cannot be stored in a cell. A clone born 10 years later grows up with a different mother, litter, and household. Even appearance can move. Pigment patterns are influenced by development and epigenetics. The first cloned cat, CC, did not reproduce the donor's calico pattern exactly, and later cloned dogs, profiled by The New Yorker, had small differences in markings and eye appearance. None of that means the cloning failed. It means the product was biological rather than photographic. The strongest company in this market has to manage that expectation before it becomes a complaint. Do not promise the same memories. Do not promise immediate recognition. Do not imply that grief will disappear when the carrier arrives. Sell genetic continuity. Sell inherited possibility. Sell a new relationship with an unusually close biological connection to the old one. That may sound like weaker marketing than bring your pet back. It is stronger protection for the business. Because the technical result can be perfect while the emotional result still feels wrong to the customer. A DNA report can prove genetic continuity. It cannot prove identity. And the more a company prices the emotional meaning of the animal, the more carefully it has to define the scientific limit of what it sells. Now we reach the part of the business that cannot be treated as a footnote. A cloned puppy requires more animals than the puppy the customer takes home. There are egg donors. There are surrogate mothers. There can be reconstructed embryos that do not develop, transfers that do not produce pregnancy, and pregnancies that do not produce a healthy birth. Viagen says every animal involved is continually monitored by trained staff and veterinarians. In the 2025 American Animal Hospital Association article, a company manager said surrogate animals produce one litter, then are retired and placed for adoption. Critics do not consider that enough. The British Small Animal Veterinary Association states that it does not support commercial companion animal cloning or commercial genetic storage performed expressly for that purpose. It cites limited efficiency, the use of multiple animals, and unresolved welfare concerns. The FDA's major risk assessment focuses mostly on livestock, not commercial pet orders. It found increased health risks for some young clones and surrogate dams, while also noting that healthy survivors can become normal adults. So, neither extreme is useful. Not every clone is sick, and a healthy delivered puppy does not erase the failed embryos, pregnancies, and surrogate burden behind the process. For the business, animal welfare is not only an ethical debate, it is a cost center and a reputational moat. Veterinary staffing, animal care facilities, monitoring, adoption programs, records, biosecurity, and credible outcome reporting all cost money. They can also create trust. A weak operator can cut those costs and temporarily improve its margin. It also creates the possibility of a single investigation, lawsuit, whistleblower account, or viral image destroying demand across the entire category. This market is too emotional and too small to absorb many scandals. The company is asking customers to believe that love, biotechnology, and animal care can exist in the same transaction. Trust is not branding around the laboratory. Trust is part of the laboratory. And this industry has already produced one famous example of what happens when public fascination is mistaken for durable demand. In the early 2000s, pet cloning looked ready to become a new luxury industry. A company called Genetic Savings and Clone grew out of a multi-million-dollar effort backed by billionaire John Sperling, who wanted to clone a beloved dog named Missy. The story generated enormous attention. The company opened a genetic bank. It helped produce cloned cats. In 2004, it delivered a cloned cat named Little Nicky to a customer for $50,000, widely described as the first commercially sold cloned pet. That should have been the beginning of the market. Instead, it exposed the gap between a product people loved discussing and a product enough people were willing to buy. The company cut its cat cloning price from $50,000 to $32,000. Demand still did not appear at scale. Public fascination is not demand. An earlier company learned that with billionaire backing, according to contemporary reporting, it produced 5 cloned cats and sold only 2 to paying customers. In 2006, Genetic Savings and Clone announced that it would close because the technology had not become commercially viable and demand for cloned cats was too small. The company had publicity, wealthy backing, and a unique product. It did not have enough customers. Curiosity did not become paid demand. Viagen survived by following a different path. It had broader experience in animal cloning and reproductive technology. It added dog and cat cloning after working in livestock and horses. It made genetic preservation the first step. It built a veterinary network. It remained, according to its own manager, a boutique service rather than pretending the category was about to become ordinary consumer retail. Then the acquisition by Colossal changed the strategic frame again. The technology no longer has to be justified only by the number of grieving owners who buy a clone. It can also be valued as biobanking, reproductive infrastructure, and conservation capability. The first company tried to build a pet cloning store. The surviving company looks more like a specialized biotechnology platform with a pet service at the front door. That difference is what actually scales. Mass production may be the wrong objective. The systems around pregnancy scale better than pregnancy itself. A veterinary network can expand. Cell culture protocols can become more consistent. Cryogenic storage, digital records, and customer support can handle more cell lines. A trusted brand can reduce the fear that irreplaceable material will be mishandled. A parent biotechnology company can reuse the same expertise in horses and conservation programs. The least scalable part remains the final biological conversion. Every clone requires eggs, embryo work, a surrogate, pregnancy, nursing, and veterinary care. That is why preservation matters commercially. It can serve more customers than the reproductive pipeline can immediately process and create a future pool that converts gradually. Store renewals also smooth cash collection, even though they reduce the later cloning invoice for customers who convert. The benefit is earlier cash, a continuing relationship, and a clearer view of future demand. It is timing and retention, not free extra revenue. This is not a software subscription. Custody of unique biological material requires duplicate storage, monitoring, records, insurance, and a plan for transfer or termination. But the commercial shape is clear. The clone creates attention. Preservation creates the customer base. Storage creates continuity. Scientific efficiency decides how many high-ticket orders can be completed without consuming the margin. We now know why a customer pays. They are paying to prevent a genetic line from disappearing and to create the possibility of a new relationship with its later-born twin. The final question is whether a $50,000 list price makes the provider rich. Private companies in this niche do not publish enough audited detail to reconstruct a reliable income statement, so the final numbers cannot honestly be presented as averages. The best we can do is build two transparent stress tests from public prices, the published credit rules, and explicit cost assumptions. These are original illustrative operating stress tests, not Viagen results, industry averages, audited margins, cash flow statements, GAAP accounting, or earnings promises. Both assume that each cloning customer has already paid an average of $3,100 in initiation and storage fees. Those prior payments reduce the remaining cloning revenue at delivery to $46,900 per project. Assume the operator begins the year with 1,000 stored cell lines. In this simplified model, 980 accounts renew storage at $150 each. The other 20 convert to cloning and are not counted again as separate renewals. Storage revenue: $147,000. 200 new customers purchased genetic preservation at $1,600 each. New preservation revenue: $320,000. The company delivers 20 healthy clones. At $46,900 of remaining revenue per project, cloning contributes $938,000. Modeled annual revenue: $1,405,000. Now apply explicit assumptions. Fixed and semi-fixed operating costs: $750,000. Serving the new preservation customers and renewing storage accounts: $110,000. Reproductive work, veterinary and animal care, consumables, failed attempts and delivery across 20 completed projects: $430,000. Modeled operating result before tax: $115,000, a little over 8% of revenue. That is not a claim about the real margin. It shows how a successful year can still produce a thin result once credits and biological work are counted. Keep the same preservation funnel and renewal base, but push 10 deliveries into the following year. Storage revenue remains $147,000. New preservation revenue remains $320,000. 10 completed clones contribute $469,000. Modeled annual revenue: $936,000. Fixed costs remain $750,000. Preservation and storage servicing remains $110,000. Because delayed orders still consume donor cycles, embryo work, and animal care capacity, assume cloning project costs fall only to $300,000. Modeled operating loss before tax: $224,000. 10 deliveries landing on the wrong side of the year remove $469,000 of incremental revenue, while much of the cost base remains. And the model is highly sensitive. Change average reproductive cost by $5,000 across 20 projects, and the annual result moves by $100,000. The customer sees one puppy. The business sees preservation volume, prior credits, renewals, biological efficiency, and completion timing that all have to line up. A successful clone is the visible product. A completed clone at a controlled cost and on schedule is the economic product. People are paying for genetic continuity, not the return of a life already lived. A cloned pet can share the original animal's nuclear DNA, sex, and many inherited traits. It cannot share the years already lived. That gap is the entire business. People pay $50,000 because they are not trying to buy an ordinary puppy. They are paying to stop one genetic line from disappearing completely and to create the closest biological continuation science can currently offer. The first $1,600 keeps that possibility alive. The annual storage fee keeps it available. If the customer later clones, those payments reduce the remaining balance rather than sitting on top of the $50,000 service. The laboratory gets paid to preserve cells, hold them safely, and attempt to create a healthy, later-born twin. It does not get to control every embryo, every pregnancy, or the personality that develops after birth. That is why this business is smaller, stranger, and more fragile than the price suggests. It is part biobank, part reproductive laboratory, part veterinary network, and part grief service. The clone is real. The continuity is genetic. The second chance is emotional. And the one thing nobody can put back into the animal is the life that made the first one irreplaceable.